Good day, everyone. We are Group C, and our discussion stems from Chapter 35 Immunity. Now, what is immunity? It is a state of having sufficient biological defenses to avoid infection, disease, or other unwanted biological invasion. In different references, it is said to be the possession of cells or tissues capable of recognizing and protecting the animal against non self invaders. Now, good points to remember are whether a foreign organism can invade a host depends on the host defense system. Target cells have receptors that bind to signal molecules that initiate the synthesis of proteins needed for an immune response or compounds to battle such target cells. A good example is cytokines, which play a huge role in signaling to immune system molecules. Now, for susceptibility and resistance. A host is susceptible when it cannot eliminate a parasite before it becomes established within the body. In the viewpoint of a parasite, it is called infective. A host is resistant when it can prevent establishment and survival of a parasite. In the viewpoint of a parasite, again, it is called non-infective. A good example for invasive bacteria is Streptococcus pneumoniae, which is responsible for pneumonia. Continuing, animals have two types of immunity: innate immunity, which is the base defense of an organism that doesn't depend on prior exposure from the parasite or invasive species within its body, and acquired immunity, which is developed immunity against a specific non-self material from prior exposure from it. Now, frequently, resistance conferred by immune mechanisms is not complete, and premonition can occur, wherein some parasites may remain and reproduce slowly after recovery of the host with clinical measures. Moving towards innate defensive mechanisms, animals have physical barriers and chemical defenses. Animals can have unbroken surfaces that can be tough and cornified as a barrier to foreign organisms, and some produce mucus that contain parasitidized cell substance IgA and lysozyme. IgA can cross cellular barriers to reach specific invaders, and lysozyme attacks the cell wall of many bacteria. In response to different stimuli or invaders, different cells liberate protective compounds such as interferons, which are released by eukaryotic cells in response to invasion by intracellular parasites, viruses, and other stimuli. Another is tumor necrosis factor, TNF, from the family of cytokines is produced by many cell types such as macrophages, T lymphocytes, and white adipose tissues. It is a major mediator of inflammation and can cause fever in certain concentrations. It is good to note that fever is the most common symptom of infection within an animal. Complements also exist, which are proteins that are activated in sequence in response to an invading organism. One pathway is the classical pathway, which is due to antibody present on the surface of the invading organism. Another is the alternative pathway, which is due to interaction of complement proteins to outer coating of the invading organism, which initiate defensive response to bacteria and fungi. Both pathways rely on the third component in the complement cascade C3, which ultimately lyses the invading cell. Active C3 binds invading cells and tags them for phagocytosis. As a quick description, phagocytosis is the process of engulfing a cell, in this case to ingest and destroy pathogens like viruses and bacteria and infected cells. Tagging molecules are called opsonins, and the process of tagging is opsonization. This is a GIF of phagocytosis of a bacteria by leukocytes or white blood cells. Other animals are observed to be resistant to many microbial pathogens, and with help of experiments, hundreds of antimicrobial peptides have been identified. These antimicrobial peptides do not have high specificity, and each peptide is effective against a different category of microbes. Antimicrobial peptides in animals are called defensins. The release of peptides begins when receptors recognize a microbial molecule. Receptors are mostly tall proteins or toll-like receptors, and their activation signals the nucleus to produce a peptide for the microbe present. Expounding more on phagocytosis, cells that do phagocytosis are called phagocytes. Lysosomes are cytoplasmic vesicles that help phagocytes by providing digestive enzymes to destroy an engulfed particle. They contain enzymes that catalyze the production of ROIs and RNIs that produce intermediate compounds that are potentially toxic to engulfed invasive microorganisms. Monocytes also exist from stem cells in bone marrow and give rise to macrophages, which are members of the mononuclear phagocyte system, which are stationed around the body. For example, Kupffer cells in the sinusoids of the liver, osteoclasts of the bone, Langerhans cells of the skin, and microglial cells in the central nervous system. There are also polymorphonuclear leukocytes, which are phagocytes in blood commonly called granulocytes, subdivided into neutrophils, which are the most abundant of all, and the first line of phagocytic defense in an infection. Their lysosomes help produce ROIs that are toxic to invading organisms. Eosinophils, which about 2 to 5% of total leukocytes, actively destroy invading parasites by releasing their granules at the site of infection and limit inflammatory response. And basophils, which are the fewest of all leukocytes, release their granules during an inflammatory response. There are also mast cells, which are basophil-like cells that also release their granules during inflammation. There are also lymphocytes, which are crucial to acquired immune response of vertebrates, subdivided into T lymphocytes, T cells, which work in cell and humoral mediated immunity of the body, and B lymphocytes, B cells, which work in the humoral mediated immunity and antigen presenting cells. Also, there are innate lymphoid cells, ILC, one of which is a natural killer cell, and K, lymphocyte-like cells, kill virus-infected and tumor cells of the body by releasing interferons, cytokines that activate other defense cells that lies a target cell, and ROR, ILC, that occurs in the intestinal lumen, which secretes cytokine interleukin 22 that restricts commensal bacteria in the intestine and stimulates antimicrobial peptide production due to intestinal pathogen invasion. Here we have visual representation of different blood cells, where in fact it includes the monocyte, lymphocyte, neutrophil, eosinophil, basophil, a macrophage, the red blood cell which is erythrocyte, and platelets. Continuing to inflammation.
Inflammation. Inflammation is a vital process in the innate immune response involved in mobilization of body defenses against an invading organism or other tissue damage and in repair of damage thereafter. The inflammatory process is greatly influenced by prior exposure of the body to the invader and by the duration of the invader's presence or its persistence in the body. The innate processes by which an invader is actually destroyed are non-specific but usually lead to activation of the acquired immune response. The immune response is how the body recognizes and defends itself against bacteria, viruses, and substances that appear foreign and harmful. The inflammatory response occurs when tissues are injured by bacteria, trauma, toxins, heat, or any other cause. The damaged cells release chemicals including histamine, bradykinin, and prostaglandins. These chemicals cause blood vessels to leak fluid into the tissues, causing swelling. This helps isolate the foreign substance from further contact with body tissues. During an inflammatory response, there is an early phase in which damaged tissue releases chemicals such as histamine, which cause dilation of blood capillaries and increase permeability, allowing leukocytes to move from capillaries to reach the damaged region. Blood plasma leaks into the region and the area swells, helping to isolate the region from potential foreign invaders. Bradykinins are also released from the damaged cells, and this causes a pain sensation. The basis for allergies and asthma is the immediate hypersensitivity. Immediate hypersensitivity occurs when mast cells and basophils become sensitized by foreign substances such as pollen and bee venom. Some scientists propose that the allergic response originally evolved to help the body ward off parasites because only allergens and parasite antigens stimulate production of large quantities of IgE. There is a hypothesis that in absence of heavy parasitic challenge, the immune system is free to react against other substances such as ragweed pollen. Therefore, people who live where parasites remain abundant are less troubled with allergies than are those living in relatively parasite-free areas. Grafted tissues from another individual of the same species is called allograft, while xenograft is grafted tissues from another individual of another species. This is used to test the ability of tissues to recognize non-self. If the graft grows in place with no host response, the host tissue is treating it as self. But if cell response and rejection of the graft occur, the host exhibits immune recognition. Evidence from studies of innate immunity in invertebrates has begun to blur the lines between acquired and innate immune systems. Although many of the mechanisms are quite different, analogous phenomena of memory and specificity of response have now been found in invertebrates. The specialized system of non-self recognition possessed by vertebrates produces increased resistance to specific foreign substances or invaders on repeated exposures. Antigen is a specific foreign substance that stimulates the immune response. The acquired immune response has two defense mechanisms called humoral and cell-mediated. Humoral immunity is based on antibodies, which are both on cell surfaces and released into blood and lymph, whereas cellular immunity is entirely associated with cell surfaces, in which the humoral immune system deals with antigens from pathogens that are freely circulating or outside the infected cells, while the cellular immunity occurs inside infected cells and is mediated by T lymphocytes. Antibodies are proteins called immunoglobulins. As what we can see in this picture, antibody has two identical light chains and two identical heavy chains forming a Y shape by disulfide bonds and hydrogen bonds. The antigen binding site occurs at each end of the Y that forms a cleft. Functions of antibody in host defense. Antibodies can mediate destruction of an invader in a number of ways. It can neutralize toxins secreted by an invader, and the binding of complement to antigen antibody complexes facilitates clearance of harmful masses by phagocytic cells. Also, the antibody bound to the surface of an invading cell may trigger contact killing of the invader by host cells in antibody-dependent cell-mediated cytotoxicity.
T cell receptors. T cell receptor is a protein complex located on the surface of T cells, or T cell lymphocytes, and is responsible for the identification of antigen fragments as peptides attached to large histocompatibility complex molecules. Subsets of T cells. Cytotoxic T cells. Cytotoxic T cells kill their target cells primarily by releasing cytotoxic granules into the cell to be killed. These cells recognize their specific antigen when presented by MHC class I molecules that are present on the surface of all nucleated cells. Their main function is to kill virally infected cells, but they can also kill cells with intercellular bacteria or tumorous cells. T helper cells. These cells secrete cytokines that modulate the activity of other types of lymphocytes and macrophages during an immune response. Th1 activates cell-mediated immunity against bacterial and viral attack. Th2 activates humoral immunity and antibody release. Memory T cells. Memory T cells are important because they can quickly expand to a large number of effector T cells upon re-exposure to the antigen and have a low threshold for activation. They provide the immune system with memory against previously encountered antigens. Memory T cells may either be CD4+ or CD8+. Cytokines. Cytokines are a large group of proteins, peptides, or glycoproteins that are secreted by specific cells of the immune system. Cytokines are a category of signaling molecules that mediate and regulate immunity, inflammation, and hematopoiesis. Interleukins. Interleukin is any group of naturally occurring proteins that mediate communication between cells. Interleukins regulate cell growth, differentiation, and motility. They are particularly important in stimulating immune responses such as inflammation. Interferons. Interferons are a group of signaling proteins made and released by host cells in response to the presence of several viruses. In a typical scenario, a virus-infected cell will release interferons, causing nearby cells to heighten their antiviral defenses. Tumor necrosis factor. Tumor necrosis factor is a multifunctional cytokine that plays important roles in diverse cellular events such as cell survival, proliferation, differentiation, and death. As a pro-inflammatory cytokine, TNF is secreted by inflammatory cells which may be involved in inflammation-associated carcinogenesis. Generation of a humoral response Th2 arm. Humoral immune response. Humoral immunity refers to antibody production and the accessory processes that accompany it, including Th2 activation and cytokine production. germinal center formation and isotype switching, affinity maturation, and memory cell generation.
And now, let's move on to cell-mediated response, T helper cell type 1 arm. Many immune responses involve little antibody and depend on the action of the cells only. In cell-mediated immunity of the epitope of an antigen is also presented by APCs or antigen-presenting cells, but the Th1 arm of the immune response is activated. In this case, APCs may be virally infected cells, tumor cells, or infected macrophages that have phagocytosed bacteria. Th1 cells recognize the epitope MHC2 complex and become activated to release IL-2, TNF, and interferon Y. IL-2 promotes the activity of activated B and T cells and enhances the cytotoxic activity of natural killer cells, causing them to proliferate and to become lymphocyte-activated killer cells or LAK cells. LAK cells also release INFY. INFY is a potent macrophage activating factor. It promotes B and T cell differentiation and B cell proliferation. It also activates capillary endothelial cells so that lymphocytes may pass through blood vessels into infected tissue areas. INFY is also a major inducer of inflammation. TNF activates PMNs and stimulates macrophages and cytokine production. Cytotoxic T cells also interact with the APC surface receptors, and this, together with stimulation by IL-2 and INFY, causes them to proliferate and to secrete proteins that cause pores to form in the infected cells, causing lysis. Like humoral immunity, cell-mediated immunity shows a secondary response due to large number of memory T cells produced from the original activation. For example, a second tissue graft between the same donor and host is rejected much more quickly than the first. And now, let's move on to the next topic, Acquired Immune Deficiency Syndrome or AIDS. AIDS is an extremely serious disease in which the ability to mount an immune response is disabled severely. It is caused by human immunodeficiency virus or HIV. HIV preferentially invades and destroys the Th lymphocytes. Normally, Th cells form 60% to 80% of the T cell population. In AIDS, their levels are undetectable, so the humoral immune response is destroyed and the cell-mediated response is compromised. HIV infection virtually always progresses to AIDS after a latent period of some years. Since Th cell populations are virtually destroyed by HIV, AIDS patients are continuously plagued by infections with microbes and parasites that cause insignificant problems in persons with normal immune responses. AIDS is a terminal disease if left untreated. Blood group antigens, ABO blood types. Blood cells differ chemically from person to person, and when two incompatible blood types are mixed, agglutination of erythrocytes results. The basis of these chemical differences is naturally occurring antigens on the membranes of red blood cells. The best known of these inherited immune systems is the ABO blood group. Antigens A and B are inherited as codominant alleles of a single gene. Homozygotes for a recessive allele at the same gene have type O blood, which lacks A and B antigens. According to the presence of A and B antigens, there are 4 types of human blood. About 41% of the whole population acquires blood type A, which means A antigen is only present. About 9% acquires blood type B, which means that only B antigen is present. About 3% acquires blood type AB, which means that both A and B antigens are present. And lastly, About 47% of the whole population acquires blood type O, which means that both A and B antigens are absent. And now, let's move on to the last topic, Rh factor. Karl Landsteiner, an Austrian physician, discovered ABO blood groups in the 1900s. In 1940, 10 years after receiving a Nobel Prize, he made another famous discovery. This was a blood group called the Rh factor, which was named after the rhesus monkeys in which it was first found. The Rh factor is encoded by a dominant allele at a single gene. Rh positive and Rh negative bloods are incompatible. Shock and even death may follow their mixing when Rh positive blood is introduced into an Rh negative person who has been sensitized by an earlier transfusion of Rh positive blood. Rh incompatibility accounts for a peculiar and often fatal hemolytic disease of the newborn, erythroblastosis fetalis. If an Rh negative mother has an Rh positive baby, she can become immunized by fetal blood during the birth process. Erythroblastosis fetalis can be prevented by giving an Rh negative mother anti-Rh antibodies just after the birth of her first child. These antibodies remain long enough to neutralize any Rh positive fetal blood cells that may have entered her circulation, thus preventing her own antibody machinery from being stimulated to produce Rh positive antibodies. The mother must be treated after every subsequent pregnancy. If the mother has already developed an immunity, the baby may be saved by an immediate, massive transfusion of blood free of antibodies.